On the left is an anti-parallel double helix structure of DNA. It is composed of nitrogen bases and a sugar phosphate backbone. Also on the right is another DNA structure. They are paired up by cytosine and guanine and thymine and adenine. They are connected by hydrogen bonds. They are also composed of deoxyribose sugar, which is the dark blue, and phosphate, which is the light blue. These hold the whole structure together.
The first step of DNA replication is when the helicase, which is labeled here, breaks the hydrogen bonds between the nitrogen bases. The reason that the helicase breaks the hydrogen bonds is for these two parental strands to stay so that new strands can come in and create a new set of DNA.
The next step is when DNA polymerase, which is right here, connects three floating nucleotides labeled here and joins them to the previously broken hydrogen bonds by producing sugar phosphate.
The DNA polymerase continues to join the nucleotides to create two strands of DNA, which will be here and here. And they will break off from the original strand of DNA, which is up here.
After the DNA polymerase does its job, the DNA ligase will the sugar phosphate bonds together, here and here.
This is the result of the DNA polymerase joining freely floating nucleotides to the old nucleotides in the parental strand. As you can see, these are the phosphates along the sides, and these are the deoxyribose sugars. In the middle are the complementary base pairs, and A always goes with T, and G always goes with C. Obviously, the DNA will be longer than this, since human DNA is 3 feet long, but this is just a small diagram of how the nucleotides connect to each other.
The DNA polymerase adds complete complementary nucleotides to the gaps, and the DNA ligase adds phosphate in the remaining gaps of the phosphate sugar backbone.
Each new strand of DNA is composed of one parental strand, which is the old strand of DNA, and one daughter strand, which is the new strand created by the DNA polymerase and the ligase. Before the new strand of DNA is completely created, errors and corrections are made by enzymes which remove wrong nucleotides, and DNA polymerase fills them.
This is semi-conservative replication. It gets half of the parental strand, which is the blue, and half of the daughter strand, which is the orange.
This is a good model to show how DNA starts off in one strand and it separates from the helicase, and then the DNA polymerase and ligase do their work here, and it ends up creating two new strands of DNA.
These diagrams are more complex because they show different parts of DNA replication at the same time.